This is a video for the BioEssays article, Why the Lipid Divide? Membrane Proteins as Drivers of the Split between the Lipids of the Three Domains of Life. The lipids of the three domains of life are drastically different. While the overall function is the same in all three domains, the lipids of archaea typically have isoprenoid chains in their tails, whereas the lipids of bacteria and eukaryotes typically have fatty acids. The link between the tails and the head group is typically an ether in archaea and typically an ester in bacteria and eukaryotes. Finally, perhaps most surprising of all, the head group is typically an S-N-glycerol 1-phosphate in archaea, but an S-N-glycerol 3-phosphate in bacteria. These are very similar molecules, but mirror images of each other, or enantiomers in the chemical terminology. These vast differences between the two types of lipids are known as the lipid divide, and understanding the causes for this divide remains one of the most fundamental unresolved questions in evolutionary biology. One of the ideas that have been put forward to explain the lipid divide is that the lipids themselves would be unstable if mixed, such that they would spontaneously segregate into the two types, either in a chemical timescale or in an evolutionary timescale. So if there were a cell that had both types, relatively quickly in evolutionary terms, we would end up with two lineages, the archaeal type and the bacterial type. However, recent results show that it is possible to mix the archaeal type and the bacterial type lipids and produce stable liposomes. Further, it is possible to take the archaeal genes that produce the membrane lipids and clone them into E. coli, such that the E. coli produces both types of lipids. The cell is actually stable to several types of environmental stress. But in spite of these successes in the laboratory with engineered strains and liposomes, it has not been possible to find archaea with bacterial membrane lipids or bacteria with archaeal lipids living in nature. Until recently, members of the bacterial FCB supergroup have been reported to have both types of lipids in nature. So there does not seem to be any intrinsic genomic or structural reason why the two types of lipids could not mix. However, they rarely do. Archaea typically have archaeal-type membrane lipids and bacteria have bacterial-like membrane lipids. So the question I ask here is, if the two types can mix, why don't they? I suggest that if it's not the lipids themselves causing the lipid divide, then it may well be the other main component of cellular membranes, membrane proteins. Membrane proteins are crucial to many of the processes in the cell, for example reproduction, bioenergetics, cell-to-cell communication, motility, transport, nutrient acquisition, waste disposal, surface interactions, homeostasis, and more. They compose over 50% of membrane surface and over 75% of the membrane by mass. They are involved in shaping the cell, and they evolve in close adaptation to the lipids with which they interact. I suggest that if the lipids themselves are not driving the lipid divide, then it may be this interaction between the lipids and the membrane proteins that is doing so. In this way, archaeal membrane proteins would be stable in archaeal lipids and bacterial membrane proteins would be stable in bacterial lipids, but if somehow bacterial lipids were to find their way into an archaeal membrane, the resident archaeal proteins would not be as stable as they would have been in the purely archaeal membrane. So in the evolutionary path of this archaeon on the right, it may acquire the genes for synthesizing bacterial membrane lipids. I suggest that if that is the case, the resident archaeal membrane proteins will be negatively affected by the import of the foreign lipids, such that the cell would not be viable at all, or it would very quickly lose the expression of the genes for bacterial membrane lipids, thus reinforcing the lipid divide. For further details, please read the article on BioEssays.